I've been, as Miranda said, and welcome everybody. It's good to see a good number of people here, and to those beyond the room. I've been thinking for quite a while now about, as Miranda mentioned, looking at how we might bring a bit more physiological insight into plant improvement, and this has been going on for over a decade, so it's not something that's thought up last week. But the main motivation for this was really, I think there's an amazing potential to add value to the rapid advances in genotyping and phenotyping, and those advances have been astronomical. Okay, so now plant breeding programs genotype just about everything they play with, and there's really advanced phenotyping technologies out there that are generating lots of information, but most of it goes into a black box when it comes to try to figure out what it means and what you can do with it. Some of those black boxes have been pretty effective, mind you, so they're not all bad. But I think we can do a lot better, and that's where I'd like to try to take you with the talk. So what I want to do is go through 5 sections, talk a little bit about genetic improvement. As Miranda said, I'm a crop physiologist and modeller, so I'm not a pre-breeder or a plant breeder, but I've been working with a number of them for a number of years, and so you'll get my view of what plant breeders do. There's probably a couple in the room who will complain, but anyway. And the notion of genetic gain and how central that is in plant breeding, and how the black box comes into that whole idea of trying to get genetic gain in breeding programs. The central role of phenotypic prediction, that's a key component of genetic advance. And I'll talk a little bit about complexity and context dependencies, and how phenotypic prediction is not straightforward when you get to complex traits. And that's where we can think about introducing biological insight by understanding crop growth and development dynamics, and how you can use that to generate genetics by environment interactions. And so that leads into targeting phenotype and GDP prediction. So if you understand a little bit about what's really going on, then you can look at what you need to measure rather than just measure what you can, and target your capability to predict around that understanding. And I'll give a couple of examples that use different aspects of that. And then I just want to finish up with a little bit about how do we go beyond this black box. And I think there are some really exciting things happening that can integrate the sort of the blunt grunt of statistics and mathematics with biological insight. And that can be used to inform approaches to future crop improvement, and I think that's already happening. So let's start off with crop physiologist's view of genetic improvement. So we've got this sort of on the left here, the simple notion of parental pool, crossing those parents, growing them out, selecting the best, and having these cycles of selection that can bring in new traits and ultimately send out new cultivars to industry. So this is a very simplistic cycle of selection view. If we move though onto a paper where Scott Chapman sort of expanded this a little bit, and you still start off with this parental pool, but you can introduce some novel germplasm. Choosing the parents to select and cross is in itself an interesting decision. And then crossing those, looking at early generation selection and phenotyping, taking that through to multi-environment testing in later generations, ultimately selecting out the best for cultivars for industry, feeding back into these parental lines and going around this cycle. And so there's a lot of statistical prediction in here, and there's a lot of phenotyping in testing as you go through these cycles of selection. There's a lot of new technologies that are now enhancing this. So I've added these onto Scott's figure. The advances in genotyping, so you can now chip seeds or do things and just get whole genome profiles very cheaply. That gives you a lot of information. It doesn't necessarily give you a lot of knowledge. There are things like double haploids where you can rapidly produce homozygous material, and so that gets you through this whole cycle a lot quicker. High throughput phenotyping, where you can get a lot more bang for your buck in phenotyping, but again, generating lots of information, and the question becomes what to do with it. And genomic prediction, where you can start to look at connecting these genotypic and phenotypic data into prediction models that say, well, what's the phenotype given I've got this genotype? And so this system is fairly conventionally, from my perspective, how I see plant breeding programs work. The focus in these, though, is really around this notion of achieving genetic gain. And central in that is this concept of phenotypic prediction. But before we get to that, the breeder's equation, this one. So genetic gain is really a product of the selection intensity, i.e. what proportion of your population do you actually maintain, keep, select. The heritability, what fraction of the phenotypic variance is actually genotypic, and yields per cycle, how quickly you go through those cycles of selection. So the quicker you go, the more rapid your genetic gain. And involved in doing this, so these are the things that you're trying to improve so you can make more rapid genetic gain as you go around those cycles. And phenotypic prediction is a core component of that, and traditionally that's been done with this model where the phenotype of genotype A and environment J is some mean with a genotype effect, an environment effect, and an interaction effect. Now that's the basic sort of fundamental equation that's then split out into whole lots of other more complicated equations that look at different sorts of variance components in each of those terms. But if we then go back to where is this black box? The black box is really in this space between genotype and phenotype. And so what we've traditionally been doing, and here's your, you've now got these things where you've genotyped all the entries in your population. Here's your population in the field taken from a UAV. And so you've got all your plots, and each of these plots is genotyped. And so you're back to using this equation of statistical connection between phenotype and genotype to determine which entities to select and progress. And so that works. I mean, it's worked forever, and it continues to work. And so it's not that it's ineffective, it's just that let's look at whether we can do better. So phenotypic prediction is where I wanted to focus then, and think a little bit about the sorts of data that we can now access. And I think I've shown this sort of picture before, where you've got sort of masses of data of genotype and phenotype now. So you can genotype everything, and this is from an installation at the Museum of Old and New Art in Hobart, where they had a sort of cascade of this data running down a wall, which I thought was rather nice. But then you can also do this for phenotyping. So now we've got these phenotyping platforms where you can run things around on conveyors and twist the plants and measure their hyperspectral reflectance in 1,000 wavelengths from every angle you might choose. And you can also run UAVs over field trials and produce point clouds of canopies, masses of data, incredible masses of data. And so the whole sort of game has gone towards getting this data. And so now the view is, well, we've got a data problem. It's no longer we've got a genotyping problem or a phenotyping problem, we've got a data problem. What do we do with all this data? And so suddenly we get this emergence of all sorts of big data solutions, deep learning algorithms, the whole works. But the whole thing is sort of like driven by what we can measure rather than why we're there. And so the point is really, how do we bring some biological insight into this to enhance the effectiveness of scaling from gene to phenotype? And that's the question that I think I want to get at here. Big data solutions and all these sort of algorithms are, again, potentially useful, but at the moment there's a targeting problem. So let's go back a bit and think about biological organisation and scales, crossing scales, and how that confounds prediction, because that's critical in here. Predicting from genotype to phenotype, you're crossing a whole lot of scales of biological organisation. and from gene through pathways to cell, organ, plant, up to crop. Now, the conventional method is to just say, okay, we understand some genetics profile, we have a phenotype at the crop scale, we've measured yield or growth or whatever, and we can fit some model, quantitative genetics or some genetic prediction model, and we can sort of connect the two ends up. But this falls apart because of the context dependencies of that prediction on A, the genotype, and B, the environment. So if you, and I can give you a very simple example, if you change the tillering of a sorghum plant, for example, and you grow it in a well-watered, high-yielding environment, the high-tillering one will yield well and the low-tillering one won't. In a droughted environment, the opposite will occur. So you've got this sort of immediate dependency of your answer on the environment that you grow the thing in. Similarly, if you grow the thing in a well-watered environment and you've got something that's high-tillering, if it doesn't set a high seed number, it won't capture the advantage it's got from the extra light it's intercepting. And so you've got a gene-by-gene context dependency, because if you don't have the extra sink, you can't make use of the extra source. And so these things confound these, the ways of doing these quantitative models. So the problem is biological models don't necessarily deal with it all that well either, because there's a long way from here to here. And so I've been working largely in this space in functional whole plant models where we go from organ to crop and back. But there's a whole lot now of gene network models where people play around with gene interactions with biochemical pathways and maybe get you up to sort of something at cellular level. But there's nothing really that scales the whole lot. It's just a little bit too complicated. But if we break this down a bit and start thinking about, well, let's think about the traits that we're particularly interested in, then this phenotypic distance isn't always this major problem, because you can have what I call a short phenotypic distance where traits scale directly from gene to crop level. And if you think of what I call relatively simple traits, they're still not necessarily that simple. But if you've got something where there's an expression of a protein that gives you a disease or a pest resistance, for example, so you've got a very clear path from expression to phenotype at the plant level, that's a fairly short phenotypic distance, and you really don't necessarily need all this crop modelling sort of stuff. And you can start to use genetic prediction and gene network models and things like CRISPR if it gets down to there's very few genes involved and you know the connection between the pathway and the phenotype. And so, but the problem is people who work at the gene level think that this model is the only one, okay? And that's the fallacy, because most often it isn't. The sort of short phenotypic distance is usually the exception rather than the rule. Normally what you've got is something that's a lot more complicated, and you don't scale directly from here to here very often. And so what happens in this instance is you really need to break it down. You've got those G by E problems, and the scaling is confounded. So you can use sort of models to help you understand the complexity and bring it back to a level where you can connect with these more conventional quantitative genetic approaches. And this is the space where we're sort of going. And what you've got to do if you think about this is the way complex traits work, apart from trying to explain this to molecular biologists who still have a single gene focus on the world, that you've really got to rethink about the way you do phenotyping and how you use the advances in genotyping. You've got to look at component traits, how they combine to give you this complexity across these scales, and how you might structure genetic prediction models to incorporate your ability to break down this pathway. And that's the point I want to get to. So if we want to introduce biological insight into this black box between genotype and phenotype, you've got to start thinking about how do you break down the complexity. And I've put models in the middle here, but it's not really the model. It's the understanding of the quantitative, the physiology that's going on and the dynamics of crop growth and development. And from the phenotype, you can sort of dissect out the components that are generating that phenotype and connect them back to genetics. And you can use an understanding then of the genetic architecture to come back to how does that link to the crop model and how do I predict the phenotype. So you're putting a prediction algorithm in here that isn't a statistical one, it's more biological. So the comment, you're getting the right answer for the right reason. And when you break that down a little bit as to what it looks like, sorry that didn't come up that well, but you know, these models don't necessarily have to be that complicated, but you've got sort of some major environmental drivers, and you really need to understand the development of the crop, its canopy development, biomass accumulation, and partitioning to grain, and how water and nitrogen and other factors interact with those processes. And normally, there's sets of equations that you can get from experimentation that help you do that. But the main point is about dynamics. The algorithms you put into this space to connect up how a crop grows and develops, really all they're doing is capturing the key aspects of the dynamics of crop growth and development. And it's that dynamics that generates the phenotype. And it's the consequence of that dynamic. Small changes can integrate to large effects on phenotype, which I'll show you in a minute. So the more complex version of the model is this one, which is actually the APSIM crop model. But essentially, it's not that different to the one I just showed, where you've got environmental factors across the top. That's nitrogen, by the way. And you have a range of processes down here, like phenological development of a crop, how many leaves it's going to produce, what leaf area will it grow, and if you know that, then you can start to look at how much crop growth rate will you get, because how much radiation will it intercept, or how much water will it use, so there's a water branch. Over here, down to the growth rate around anthesis, giving you grain number and allocation to grain size, so you get through to yield. So you can sort of break down the crop growth and dynamics into a few interlinked processes with this sort of spaghetti diagram that just really, underneath this is just a key sets of equations that enable you to link all those things up and make those predictions. So just to give an example, there'll be a test on that figure after the end, by the way. If we look at, I mentioned tillering before, and let's just say, let's reduce tillering in sorghum. So mostly I work on sorghum, so my examples are going to be sorghum. And this is just a simulation, and this is plant available water in the soil, okay? And I've said, right, let's run two plant types here. The orangey one is a standard plant type, and this is its leaf area index. And then I say, right, let's take half a tiller off that plant. And this is the leaf area index of the lower tillering one, and the red is its water use, and the blue is the water use of the standard one. And you can see the standard one grows more canopy, as you think, because it's got a little bit extra tillering. But what happens is it uses a fraction more water. It's not much, okay? It's like 5 millimetres or something, hardly measurable. But what the model will then do is say, well, okay, this one that's produced less leaf area, actually it just takes a little bit longer to get into stress, and so its leaf will stay green longer, and you get what would commonly be called in plant breeding parlance, stay green. Okay, so that type stays green longer. So what you would see in the field, if you had these two plots next to each other, is this one would have green leaves at this point, and this one would have fewer green leaves at that point. And plant breeders rate those things and then go and do genetic analysis of them. And so you can generate these sort of emergent properties of complex traits. So tillering is actually generating stay green, and that's a long way back. Tillering happens back here somewhere, okay? So if you go out and start doing sampling because this is when the leaves are dying, and you do RNA-seq or whatever you want to do, and you can spend your life chasing, you're actually looking at the wrong time in the wrong tissue, okay? So that's not going to take you anywhere. What you should have been doing is looking at the buds down here, and I don't know if Christine Beveridge is here, but it's probably something to do with sugar signalling. And if you run that single season, and this was a terminal stress to make the point, but you can then run all historical seasons, so each point on here is a single year simulation. And this is the yield difference against the yield of this standard type. And you can see in these lower yielding seasons below about 4 to 5 tonnes, you get a significant advantage, but you get quite a significant disadvantage in the good years, and that's that G by E. So you can generate that sort of stuff if you know that tillering is an important component that can do that. So let's look at this stay green example. I've already given you one of the ways we can get it. And think about targeting phenotyping and go a bit more into this G to P prediction. So first, there's some nice work that David Jordan's group have done to just make sure I don't zoom over time here. Looking at stay green over a large number of trials and years, started way back with Bob Hensel. And so here's a stay green plot, and here's a senescent one, and you can sort of see the difference. You know, this is still looking pretty stressed, but that's just about dead. And this one will yield more, and it won't fall over as much. And if you do lots of trials and analyse them, as David Jordan did here, this is the mean site yield between 2 and 10 tonnes, and this is the yield change per unit of stay green. So the positive ones over here are saying, if I've got types that stay green, and so you can go and rank that stay green between 1 and 10, then they will give yield advance, and that happens in these low yield environments. In the high yield environments, you've got a bit of a mixture going on. Some are advantageous, some are disadvantageous. But it's a very good trait in our environments because most of our environments are down this end of this picture. But let's look at how you might get stay green rather than just having this sort of generic complex trait that you can sort of see. There are a number of ways that this can happen. And Erik van Oostrom did this really nice paper where he just put together, let's look at plausible mechanisms around water that might generate stay green. And you could change the root architecture that maybe gave you a bit more water. You could change the tillering and the leaf size that changes the plant size and maybe reduces the water use at anthesis, which was that tillering example I gave you. You could change the transpiration efficiency with stomatal regulation. And all of these things give you effects on water availability at anthesis, which ultimately can flow through to increased grain yield and grain number and yield. And so it's all about changing some of these things that the dynamics then generate the effect on the water balance through the season, which change the crop growth and development dynamics, which generate a stay green phenotype and yield in those environments. And Andy Borrell's done some really nice work with experimental studies that have sort of backed up a lot of those mechanisms using near isogenic lines. So I just want to look at one of those, and this is the work from Vijaya Singh on root architecture. And so you can sort of say, well, if you've got that sort of notion, let's look at root architecture. And one of the things that she did, I'll just say there, we looked at nodal root angle and root system architecture and the ability of those root systems to capture water. And these are just images from 2 divergent genotypes with a narrow, these are nodal roots in sorghum that frame the architecture of the mature plant root system. These boxes are 1.2 metres wide and 1.2 metres deep. And so you can see the ones with the wider angle of the nodal roots don't fill the space nearly as well, probably don't get quite as deep, but certainly the main effect is on the root distribution at depth, which gives this one a little bit more capacity to take up water at depth, which can be critical. So given, and we did experiments to measure all that and to sort of validate that that was happening, but you've then got the opportunity to target your phenotyping because you know you've got a trait that's changing water capture, so you can develop the root phenotyping system that does that. Well, instead of looking at stay green or yield, let's come back and look at a trait like root angle, which shortens that phenotypic distance. And so you can build targeting your phenotyping with platforms. It's much bit more difficult to do in sorghum than the ones that people have been doing in wheat and barley because you've got to grow the plants for 3 or 4 weeks at least because we need to look at nodal root angle, not seminal roots, and it takes a while for those to appear. But you can then run thousands of plants, okay? It's still not what you'd call high throughput, but you can do a lot and you can use imaging to capture these things because you're just phenotyping to measure that one thing. And when you do that, you can run it on structured populations on either from the breeding program, pre-breeding program. So either, you do the parental lines and then you can see which ones diverge in the trait, and then you can do mapping populations, nested association mapping populations, and you can start to identify the regions that are controlling just the root angle. And then we know the root angle has these other effects that will, in the dynamics of the crop growth and development, will generate stay green and yield differences in specific environments. So you can then map that back onto the breeding trials. So there's all these multi-environment trials in the pre-breeding program all around the country. And so if you know you've got a region that's controlling that, you can look at how it relates back to the performance and see if it is actually telling you something. And you can also see if it connects with candidate genes in other species. And so Vijaya did all that, and with Emma Mace helping significantly on all the genetic end of it. And there are 4 regions that explain much of the variation in nodal root angle. This picture just shows that they're quite separate from QTLs for shoot vigor. So it's not just a vigor thing. It's actually an independent trait within the roots. They co-locate with known QTLs for stay green. So people who have phenotyped for stay green and done the similar analysis, some of the QTLs for stay green line up with these. And when we do that analysis to say, well, let's look at these regions and how they, because all the testing experiments are genotyped, you can say, let's take those regions in some of the recombinant inbred lines that have been put into hybrid testing, and you can get associations between presence of those regions and yield that are significant. So you've got a validation of the fact that this is an important trait and that yield was increased in the presence of the narrow root angle allele in those breeding trials. And so what's happening now is we're progressing that even further, looking at phenotyping a large number of plants in those nested association mapping populations and chasing the genetic architecture. And one of the initial findings from that is already that one of the regions in here aligns closely with the GRO1 gene they've recently reported in rice. And so you can start to look across species and get to a level of gene discovery and just how this trait is actually manifested. So we can then simulate that, so we can go around the loop here. So instead of just working out how it works, once we've got some idea of how it works and what effect it has on water balance, we can simulate it, and we can put these things into this spaghetti diagram, which we did with tillering. We can do it with TE, we can do it with roots. So you can make some bold assumptions about how this works, but at least gives you some idea. And so the same sort of pictures as we had for tillering. This is water content of the soil, the first 2. The narrow root one is the red one. And what happens is we say, well, let's give the narrow roots 10 millimetres more water at depth. So below a metre, they will have 10 millimetres more water. And again, what happens is the green here, it just takes longer for that type to run out of water in this terminal stress environment. So you get emergent stay green as a phenotype. And again, this is in a very terminal stress to make the point, but that's when stay green happens. And here's the biomass accumulation and the yield. So the red and the green being the one with the narrow roots. And so the only thing we've changed in that simulation is the root architecture, and the way we've changed that was just by saying, let's assume that gives you 10 millimetres more water. Now 10 millimetres is something, if you ever measured soil water, it's a really difficult thing to actually see. It's almost within your error. And yet we're getting differences of a few hundred kilos of grain, and often it's more than that. So you can run that across all seasons again, and you get this same sort of pattern where you've got 5 to 10% yield advance in these lower yielding environments and not much effect up here. And so both of these, the tillering and the roots one, sort of reflect the observations that were found in those trials. The point is then you're now in a position to predict new phenotypes, because you can start combining what you're observing in high throughput phenotyping and the genetic architecture of it, and put that into models that say, well, it would be nice to have this combination of regions for this environment, because that would generate this combination of tillering and root architecture that would be advantageous. So you can start to think about design. The other example I want to give is a little bit about going the other direction. Instead of thinking from a trait and dissecting it and understanding it and coming back up again, let's look at breeding trials and try to analyse them with a little bit more biology, and in terms of understanding the plot-to-plot variation that's observed in breeding trials, and rather than just allowing that to go into error terms. So the idea here is to look at breeding trials and try to remove some of the effects of things we think we know about. So we know about maturity, we know about tillering, we actually know about root architecture. So all of those entries in those breeding trials have some traits that we know, we understand, and we can predict the consequences of the variation of those traits. And so we can start to think about, well, let's take the effects of those knowns out of the trial results and look at the residuals, and then see whether we can do better genomic prediction on the residuals than on the original piece of data. And that's where this is sort of going. And the way this works is with some of this new phenotyping technologies. So you've got field-based platforms, and these are now being set up in the pre-breeding program in sorghum at Warwick. So there's sort of these rig-based sensor systems, UAV sensor systems, and data analysis pipelines that are being set up to capture all this data and convert it to the variables that we need to do this extraction of signal from each of the plots. So just an example here. Here's an enhanced image of a plot, and then one can, and this is where the machine learning stuff comes into its own. It can start to find the heads, okay? And then you can count them. And so you know how many you've got, you can do a reasonable estimate. And so this tells you, given that you know how many plants there are there as well, which you can get from an image earlier, you know how many tillers you've got, okay? And so you know how many tillers you've got, you've still got to have something to do with flowering time, which is still difficult to get from some of these images, but that's where this is going. A lot of people are going into far more detailed variables than this to try and make connections. I think there's still a lot of low-hanging fruit that can be got out of this before you worry about hyperspectral reflectance patterns across hundreds of wavelengths, and which you're not really quite sure what they're telling you anyway. So what we've been doing with this is, this poor PhD student, and has been roaming around all David Jordan's breeding trials and seeing if we can do this. So the idea is, within those trials, we have a couple of check genotypes that he runs across all these testing in sites. At those sites, you will measure biomass, leaf area at anthesis and maturity on just those checks. So you actually go in and take some quadrats, okay? So you measure some things. It's just not all virtual stuff, because we need to ground the model somewhat, because we don't know. We know we've got a weather station on the site, so you know the weather. The soil moisture is the hard bit. You don't necessarily know too much about the soil. It's very hard to characterise it really effectively. So if you've got information like this, you can always use that as a bioassay through a model to estimate what sort of soil moisture must have been there. So you're not using the model to predict anything. You're using the model to say, well, what was my boundary condition for this field experiment? Then you can look at using that to set up this notion of a template. So you said, well, okay, I know what the boundary condition is, and I know I've got hundreds, thousands of genotypes here. Let's assume they're the same as these ones, but I know their flowering time and I know their tiller number. If I run 1,000 virtual genotypes, just changing that, then all I'm doing is saying, let's assume I've got the check, but I've got one that either flowers earlier with more tillers or earlier with less tillers. I know those two things. How much variability does that explain? And so when you do that, you can sort of try to get some idea of, this is a predicted yield, not of any specific genotype, but of your check with those two things changed, okay? And so you can look at how well that relates to what's observed across all your plots and remove these confounding effects and calculate the residuals, because there will be other things in those genotypes other than flowering and tillering that are generating yield differences. It's not just those two things, but they're the two things that we think we're quite confident we can explain. And it's sort of a complicated picture, but the first analysis that he's done, here's the observed yield, and this is flowering date, and this is a particular trial on the Downs. And so there's a lot of variability in yield at each flowering date, and this is sort of some fit of the response of that. If we take those checks and we determine what we think the soil water was at this place, and we simulate that, and this is the flowering date here, and then these streams of points you can see here are where you've either got 0 tillers, 0.1, 0.2, up to about 1.5 or something. So in each of these flowering time windows, we've done a simulation of, let's increase the tillering and keep the flowering time the same. And so you can see in these, and this goes from 54 to 62, which is about here to here. So at least the sort of pattern is reflecting what's observed in the field. But what you can then think about is, okay, if I'm at this flowering date, this flowering time, then something that's got more tillers would probably give me more yield. And I would expect that. So, whereas over here, something with more tillers is going to give me less yield. And so I would expect that. So if I go back here, and instead of saying, oh, well, I just want all the ones that yield the best, you don't really, because down here you say, well, actually I'd be better off if I had something that tillered more than this. And so you can look at the residual from this and say, well, okay, you're actually a high tillering one up here, so I would expect you to be up there. But there's a low tillering one that's up there. And so that's telling me it's probably got something far more useful than the high tillering one that's up there, because it's further away from where you would expect it to be. So the residuals start to give you very targeted information. And so you can start to do some pretty nice things with how you go about enhancing genetic gain. So just to finish, I want to come back to how this might really relate to moving beyond the black box. And I think this is sort of happening. It's been happening for a while. But it's about thinking about how to use these new technologies in an integrated way rather than individually. And that's it seems to be a big challenge for people to get out of their own black box. And so everybody's got their model about how they think this works. but really there's power in each of those perspectives, but there's a lot more power when you think about how to put them together. And I think that's where the sort of dynamic modelling framework comes in, either just in helping you understand that and discuss it, but also in potentially some powerful prediction technologies. I don't want to go too much into this, but this is the sort of notion from the work of the group, Messina, Cooper and Co. Pioneer. And it's really important that this sort of virtual world and the analysis system is really closely connected with what's going on in the real world in experimentation. And I think that's another point that, this has got to be grounded in real world plant improvement programs. You can then start to, once you understand things a bit, these are simulations of trajectories of breeding programs. So you can start to play seriously with simulation of selection strategies and with some credibility. You're still restricted in the number of traits you can play with, but you can then look at different genetic architectures and where different selection strategies would take you when you change the genetic architecture of particular traits around. So I haven't got time to go into that, but that's where this sort of stuff is going. And really, it's about bringing those capabilities together to really enhance the power of adding value to these advances in genotyping and phenotyping through better phenotypic prediction. And there's a program we're involved in now in the US, and I've stolen their graphic because I sort of like it. And it's from this US Department of Energy, and they've got this program going called ARPA-E, Advanced Research Projects Agency for Energy, out of Department of Energy in the US. And they were putting out lots of sort of transformational project concept ideas, and they would get people together and try to design these things. And I got invited to the one where they were designing this, and they sort of say, okay, what should we be doing that actually connects better to improve field breeding with all of these technologies that are floating around out there? And so this discussion that I'm having with you now was one that was had in that room, and they then set up a number of huge projects to go and do it. And we're involved in one or two of those, but of course most of the money stays in the US. And they want us to help them do the modelling. But all the other stuff's going on. But the thing that they still sort of miss is the bit, it's all like, oh, let's take these technologies and somehow translate them into improved field breeding. I mean, the breeder's not an idiot. And so, the breeder out here has as much involvement, probably more so, in how these things should connect in than the experts in each of these fields. And so I just thought they missed a bit on their picture. But where this is then going is there's some papers coming out from the Pioneer crew, and where they're putting together crop growth models with whole genome prediction. And so they're looking at, they're using advanced Bayesian approaches to bring crop models into their whole genome prediction architecture. But there are other ways to do that. And so the residuals way that we've talked about is probably our more low-tech way of doing it. But this thing is happening, okay? So instead of just thinking about, phenotype equals mean plus genotype plus environment plus G by E, you can shove a crop model in there. And you've still got to do the analysis, but you can do the analysis into the parameters that really capture a lot of the context dependencies that generate those emergent properties that you can't capture otherwise. And so there's huge opportunities to do this. And in fact, if we had have put a small percentage of the money that's gone into this and some of these other things into this, we would really be zooming. But sexy stuff is here and here and in here. People don't sort of think well, unless you're out there in a breeding program, you don't really think about how do I really use this stuff to get more rapid genetic gain. But there's amazing opportunities to do that with that integration, and I think that's where we need to go. So I just want to acknowledge all the people who are involved in this. There's probably a slab more of them than that. And we've had good support in linkage projects with Pioneer. This is a European project that was also connected into this. GRDC, the Gates Foundation work, ACR. So they're probably the lead people, but there's a large number of people. Thanks very much.